Okay, so today we're going to be talking about neural communication, specifically how that happens by using action potential. So working together. So your body is made up of individual specialized cells and organs, and each of them have their own function, but they need to be able to communicate with each other to respond to the needs of the body and to stimuli, both internal and external stimuli. This is done in two ways. This is done either through hormonal or neural communications. Hormonal communications are going to be the endocrine system. Neural communications are going to be the nervous system. In the diagram on the right, you can see those two systems. The nervous system is going to be electrical impulses. This is very fast. And then the endocrine system, which is more long. So what the hormones are being released in your body for are going to be for duration, not necessarily speed, but the nervous system is trying to get a message to an area of the body. Excitability and membrane potential. So your membranes have a separation of positive and negative charges across the membrane of cells. These are controlled by mostly sodium and potassium. And this creates a membrane potential. So fluctuations in this membrane potential create electrical signals. Your body then uses these electrical signals to receive, process, initiate, and transmit messages. So as you can see here, the extracellular space has more of those sodium ions, and the intracellular membrane space has more of those potassium ions, which is going to be very important when we're talking about exactly how messages are sent in that action potential. So polarization, depolarization, repolarization, and hyperpolarization are the four types of changes in membrane potential. So polarization, the charges are separated across the membrane. The membrane potential is different than zero. Normally this sits at about negative 70 millivolts. So that charge, that negative, is associated with the voltage that represents the inside of the membrane. So the inside of your membrane is always a little bit negative. And this is known as your resting potential. Then you have what's called depolarization. So the membrane becomes less polarized. So the inside of the cell becomes more positive. Usually this is done because channels open, you have a movement of sodium ions, and it creates more of a positive on the inside of the cell, which changes that membrane potential. Then you have repolarization, which is where it's returning to its polarized state or its resting potential, and then hyperpolarization, where it actually becomes a little bit more polarized than its resting potential, more negative, so more negative than that negative 70 on the inside of cell. But eventually that resting potential does even out and it becomes polarized in its normal polarization again. And on the right, you just have a graph of what that looks like over time and the different millivolts that the membrane potential is hitting. So a triggering event. So then there's a triggering event that causes this to happen. So it alters the membrane permeability and allows a flow of ions. There's different triggers that you can have. So you can have a voltage gated channel that would be in response to a change in that membrane potential. You have chemically gated channels. That's when a chemical messenger is going to bind to a surface receptor. You have mechanically gated channels. This is going to be in response to a stretching or deformation. And then you have thermally gated channels, which are in response to temperature changes. The number one we're going to talk about here with this is going to be the voltage gated channels in response to a change in membrane potential. So action potential. So what is that? So this is a brief rapid change in the action potential, about 100 millivolt difference. It occurs at a small portion of the membrane, but then it propagates through the entire cell. These are long distance signals. So there are signals where it just kind of has a depolarization, repolarization in a specific area, but doesn't really do much. This is something that's going to be sending a message. So this could be something as far as from your spine all the way to your big toe. It's trying to get some type of message across to a different part of your body. So it's a slow depolarization until there's a certain threshold potential that is hit. Once that threshold potential is hit, then there is a sharp spike and depolarization and repolarization. So spike of both. Very quick depolarization, very quick repolarization. And these spikes are what are known as the action potential. It only lasts about 1 millisecond in neurons. And again, on the right there, you can see you have that after hyperpolarization until it returns back to its resting potential. So the sodium and the potassium channels in action potential. So an event triggers the depolarization to start. And what happens is the sodium channels slowly begin to open. So remember, normally there's more sodium ions on the outside of the cell membrane. So as those channels are opening, the sodium channels, the ions are entering to try and create that equilibrium, to try to even out between the two sides. As more ions enter, more sodium channels are then triggered to open. Again, once that threshold is reached that I just spoke about in the last slide, the remaining of the sodium channels open, which is what causes that explosion of sodium. So the full activation of the sodium also changes the conformation so that the sodium pumps are now able to be inactivated. So as quickly as they're all thrown open, they're now also closed. It's enough time that enough sodium gets through to cause that spike, but then this is also when the potassium channels now begin to open. So again, potassium was on the inside of the membrane. As soon as those channels are thrown open, the potassium starts leaving the cell. So what that does is it starts putting the membrane potential back towards negative as the positive ions are flowing out of the cell, and it's going to restore the electrical balance. And then the sodium potassium pumps will return these ions to their correct location over time. So you want there to be sodium ions in the inside, you want the potassium ions to be on the outside. The first thing that's more important is to get that charge back in order, and once that's done, then the pumps will return these ions to their correct location. So this is a look at what I was just referring to. So you have the depolarization, which is the sodium influx. So you see that on the left there. The activation gate, as sodium comes through, rapid opening is triggered. But as that rapid opening is triggered, you have this inactivation gate that now closes that pump, and it's now not able to get through. But then at the same time, it also triggers the potassium channel to open. All right, so action potential propagation. So you have this action potential is created. Now it's going to start flowing and moving so that it can send the message to where it needs to go. So the first thing we need to talk about is the structure of a neuron. So you have the dendrites, which are these extensions. These are going to carry signals towards the cell body, so signals that they're receiving from outside of the cell. The cell body is where the nucleus and the organelles are housed. And then that actually attaches to the axon, which is a single elongated segment that will propagate the action potential away from the cell towards where you need that message to be going. So the dendrite and the cell body have receptors that are going to bind these messengers. So those two together, the dendrite and the cell body, are kind of known as the cell input zone. And then that axon is going to go down to nerve endings, which are the axon terminal, which is where it's going to deliver its message to the next cell. All right, so dendrite and cell body do not contain many sodium voltage channels because this is an area that you want to be receiving a stimulus. So if there's lots of sodium voltage channels, that means it's more sensitive to creating an action potential, but we don't necessarily want an action potential created unless the stimuli is producing a strong enough response. So the dendrite and the cell body don't contain many sodium voltage channels, but if the area receives a stimuli that can produce a strong enough response, it will reach to the action potential possible areas, which are more towards that axon. The axon has many sodium voltage channels to be able to propagate the signal to the next cell. So the depolarization is by current flow. So you can kind of think of it as a wave at a stadium when all the people stand up, or lighting a fuse. Once you start it, you don't have to continue lighting it as you head down the fuse. It's already started, and it's just going to kind of take off on its own. So each action potential is going to set off the next action potential down the line. You don't see it going backward, so an action potential isn't going to spread, you know, both forward and backward, because the area that just had the action potential is actually in a refractory period where it cannot be stimulated again. And then just some other considerations. So this is an all or none response. So if the stimulation is not enough to trigger that action potential, so if it doesn't get that threshold, then nothing is going to happen. So that's, you know, an off, that's a none response. But if it does hit that threshold, the action potential is the same intensity every single time. So it's either happening or it doesn't. So, you know, how can you tell if you're touching something that's warm versus something that's hot? So the stimuli strength is going to be determined by the frequency of signals. So here you have that example on the right, a diagram of that. In the bottom on the stimulus voltage, you notice the first voltage isn't strong enough, it doesn't hit that threshold, so nothing happens. That second stimulus does have enough voltage to cause an action potential, so it causes a few action potentials. And then again, the stimulus is a stronger stimulus, so it's going to be creating more action potentials, but they're the same millivolt change that you're going to be seeing. And that is action potentials. And these are the references that we use to make this presentation.